This UCSD TV program is a presentation of University of California Television. Your support makes UCTV's programming possible. Contribute online at uctv.tv/support. Check out the YouTube original channel UCTV Prime at youtube.com/UCTV Prime. Subscribe today to get new programs every week.
Among the oldest questions of humans are the following. Who are we? What are we doing here? Where did we come from? How did we get here? And where are we going? Two of these questions are now tractable for scientific inquiry. And they are, where did we come from and how did we get here? And this subsumes the subject of anthropology, explaining the origin of humans. Our mission statement is as follows. We use all rational and ethical approaches to seek all verifiable facts from all relevant disciplines to explore and explain the origins of the human phenomenon. I'd like to go back more than 50 years when C.P. Snow pointed out that there were two domains of academia, humanities and sciences, and they seem to have a difficult time getting together. Well, it's not very different from the ongoing nurture-nature debates, and for that matter, it really comes down to culture and genes. Now, if you want to pursue anthropology, you need to avoid such false dichotomies. So we need to look at the humanities and the sciences, study nurture with nature, and the theme of today is culture-gene coevolution.
Well, I'm very grateful to be here and for the opportunity to engage in this interdisciplinary conversation. I'm very humbled to be here and hope that I have something useful to add to the discussion. I'm not a geneticist, so I'm dealing primarily with the origins of culture, in a sense. So we all toss this word around, culture, and in general it's being used to apply beyond humans to learn behaviors that differ among different populations of a species. So great apes have culture now, orcas have culture, birds have culture, lots of things that we never imagined would be included in this term have culture. But lots of my anthropological colleagues would reserve the word culture for humans. They're very uncomfortable with extending this to great apes. And it involves a much more cerebral version of culture that includes shared beliefs, shared values, and symbols. So it includes, in a sense, their definition and implies language. So one of the things we're looking at is the transition from an ape version of culture to a human version of culture. And we also have to, as the introducer said, to realize that humans not only are dependent on culture, but we're also shaped by culture. That our behavior and our morphology have evolved together in a feedback relationship. The fact that we chose to consume tubers when things were bad, we're going to hear a little bit more about this later, created set up a selective situation which selected for more genes that help the digestion of starches. And the fact that we kill and process animals that were bigger than we are, at least we did so for 99% of human evolution, also shaped other aspects of our morphology to be able to do it better. Our weapons evolved, and so did our shoulders. Our stone tools evolved, and so did our wrists to make them better. And we became better communicators with each other, and the speech apparatus and the brain both evolved to facilitate that. So we've come to the point where we can't survive without culture. You leave a human on a desert island and give them no possibility of making anything to help them survive, they wouldn't survive very long. And we have, in order to acquire that culture, we have an extraordinarily long period of dependency relative to the rest of our life cycle. Not just infancy, which many animals have, but a juvenile period, which is normally a very dangerous period for a species. And I would, in moving towards the end of human evolution and the appearance of our own species, Homo sapiens, I would underline three important aspects of our own culture. One is our capacity for technological and economic innovation, our ability to build on past innovations, to ratchet up what our forebears have done, to build something new, and to accumulate culture. This is sometimes called cumulative culture. We also have extraordinarily large social groups that constitute imagined communities, because if you think about who your family is, there are people in your family, I'm sure, as there are in mine, who you've never met. There are people who are not even alive while you've been alive that you think of as your family. There may even be, in some cases, members of families who are spiritual beings or who are going to be born in the future. And the same goes for communities or nations. All of these things have an imagined, a mental aspect to them, not just a physical on-the-ground aspect. And these large social groups are essential for faithful, long-term cultural transmission of complex behavior. They also increase survivorship because there are more individuals that you can go to for help if things are bad where you are or in your life. And the third aspect is the world of symbols, which reifies these social groupings and increases the potential for information sharing and also helps survivorship. And there are lots of ways of trying to understand the past. The way I try to understand the past is by the direct evidence that it leaves behind, which, as you see, includes at the bottom genetic inheritance, which I think is a direct proxy of what happened in the past. But so are the stones and the bones of the archaeologist, the landforms and sediments where we find the stones and the bones, and the biotic and chemical environmental proxies that are encapsulated in those sites. So the question is, for me, when does modern human culture begin, and was it a gradual or a rapid transformation? If we look at the end of the Pleistocene, we find that the first Homo sapiens in Europe had many of these, or if not all of these characteristics: material and economic innovation, symbolic culture, language, large social networks. They had flutes, they had strange sculptures that looked like half human, half animal, and so they lived in a rich symbolic world. Before 36,000 years ago. Chauvet Cave is another example, and a beautiful example of some of this imagery and symbolism that we see. But it's not just in Europe. I have colleagues who think that you had to have cold weather and to grow the symbolic brain to do these things. But we also have finds from Africa. This is a little bit less well preserved, but you can see that it's got human legs on the right and it's turning into an animal on the left. So we can be sure that this capacity for making these images did not develop in Europe. It came into Europe with the modern humans. And if we go back further into the past, we see that in South Africa, Namibia, Tanzania, Ethiopia, Senegal, and the countries of North Africa as well, we find this kind of rich symbolism that implies fully human language. We have beads, we have geometric designs in 2 different forms, or 3 different forms. We have them on bone. I don't have a picture. This is on this little slab of ochre. There are 2 of these. And then we have a series of geometric designs on ostrich eggshell that look very much like the eggshell in the center that was collected in the Kalahari Desert in the 1960s. They're around the hole that was made to use the eggshell as a canteen. And there are 16 or 17 different motifs that repeat themselves through the collection of the eggshells from Deep Kloof. So these may be individual styles or group styles or family styles in some way, but they're very distinctive from each other. And these beads are not just at a couple of sites, but this is a map that we made for a paper on African climate and the development of early modern human culture. You can see that there's this great density of dated sites in South Africa. There's quite a density in North Africa, but less, and East Africa is being filled in. So there's still a big concentration of the record in South Africa. That doesn't mean that that's the place where most of the people lived. It's the place where most of the archaeologists live. But about 10% of these sites have beads. So beads are not a rare phenomenon. They're at lots of the sites. They go back to about 105,000 years ago. So already we're back into a much earlier time period. We also have, between 100 and 60,000 years ago, point styles that differ as you go around Africa. And this could be argued to be a regional tradition that is shared among a large group of people. And they preferentially use everywhere exotic raw materials they had to go somewhere to get, often 150 to 200 miles away, which implies they're not necessarily going there themselves. There may be some kind of exchange networks going on. But if you think about the problems of exchanging things with people that you never met, if you were a chimpanzee, they would kill you as soon as you came over the horizon. So you have to come up with a way of telling them that you are a friend as you walk into their territory to get some of their stone. And we also have sophisticated hunting technology between 100 and 60,000 years ago. Bone harpoons in the lower left for fishing from some sites that we excavated in the Congo. Stone points from Blombos Cave in the upper left, which were hafted as spearheads. And then microliths from a number of places. These are from Tanzania. There are a lot of them from South Africa as well, that were hafted onto what we are pretty sure were arrowheads. So these people had, at least by 60 and probably much earlier, bows and arrow technology. So that would have allowed them to wipe out the game or the enemies at a considerable distance, rather than having to walk up to something and stab it with a spear, which necessarily is a little dangerous for yourself. We also have burials. I showed this one from Kafsa because there's a better picture of it, but there's another one from South Africa with a shell pendant that's about 90,000 years old. So these are burials. The distinctive thing about them is that they have grave goods. They were giving these dead people something, some kind of special treatment to take with them into another life, to provision them. Sometimes these things are food or beads or ochre to make them look as if they're alive. There's some kind of ceremonial, elaborate symbolism going on in these burials. Eurasia is very different. Neanderthals, 60 to 100,000 years ago, had no personal ornaments. They had very little comparable artifact diversity to what we see in Africa. They had no engraved geometric designs, and my colleague Francesco D'Erico cites some things, but I think he himself would recognize that the objects, the engraved ostrich eggshells with the geometric designs, are far more complex than anything we see in the Neanderthal sites, which just could be interpreted as scratches. There's little evidence of large social networks. Most of the stone is coming from local sources, and there are no burials with grave goods. There is a personal ornament, which I show here from Cueva Anton in Spain, dated to 50,000 years ago, when the modern humans are already on the periphery or in Europe. And there are several sites with bone and tooth beads that overlap in age with the ages of the earliest European modern humans. So whether the Neanderthals are actually making these things, or whether they're developing them in imitation, or whether they're so stressed out that they're finally getting with it and inventing symbolism, is an open question. But in any case, it's a very late phenomenon. The Neanderthals, as you see on this map, lived in Europe and into Central Asia. And we've recently become aware of another group of archaic humans who apparently lived in East and Southeast Asia, although the only remain we have of them is from a place in Western Siberia. Now, Western Siberia might be a lovely place today in a big interglacial. It was not a great place to live in the height of a glaciation, so it's very unlikely that this is the homeland of the Denisovans, as they've become to be called. Denisova itself has stone tools similar to those of Neanderthals, and it also has a Neanderthal remain. These Mousterian tools are replaced by tools of early modern humans. And again, there are colleagues who think these early modern humans tools were made by Denisovans. Seems unlikely because they look like Near Eastern tools, and Denisovans don't go that far west. But this is more likely the northwest edge of the Denisovan range, not the center. If we go to South China and Southeast Asia between 100,000 and 30,000 years ago, the tools look like the tools of Homo erectus, a much earlier ancestor. They're very simple, Oldowan kinds of artifacts. So Africa before Homo sapiens. So we've already realized that by 100, 110, 130,000 years ago, humans were pretty modern. And the oldest human fossils attributed to Homo sapiens are between 200 and 160. What about before that, the Middle Pleistocene? This marks a period of major climate variability, which differs by region within Africa. It's more severe at the top and bottom of Africa and less in the middle. But before Homo sapiens, we now have evidence for hafted weapons, for large quantities of ochre, which was being processed into powder, presumably for painting something, your face, your body, your clothes, whatever. We have long-distance networks of where raw material is moving. Almost all African fossil brain sizes after 500,000 years ago are 1,200 cubic centimeters or larger. That is, they're in our range. And we used to think they didn't do anything special, but that's because we were looking at the Europeans. And they had Levallois blade technologies, which many cognitive and archaeological scientists have argued imply sophisticated sequential action, conceptualization, teaching, and very likely language. It's a very difficult technique to learn, and it involves conceptualizing the final form of a piece on a core that you specially prepare to produce that final form. Okay, so there are 5 places in Africa where these long post-Acheulean sequences have been explored, and I've worked in 2 of them, the Middle Awash and Alorgesailie. I'm going to talk for 3 minutes about Alorgesailie. This is a site we finished excavating last summer called Alorgesailie BOK2. It has 5 separate horizons of occupation, and it's under a tuff that you can see on the left is dated to 340,000 years ago, more or less. The date is not fixed totally. We might get a surprise, but repeated samples have confirmed it. And every black spot you see on the floor in this picture is a piece of obsidian. There is no obsidian in the Alorgesailie Basin. And the obsidian here, this is a scale at the bottom left of 28 or 30 kilometers. The sources from BOK2 come primarily from the south, about 30, 40 kilometers away, and from the north, about 60 kilometers away. And by the time you get to the north, you're getting uphill into better watered kinds of environments. But if we go to a slightly younger site that's still probably older than 300,000, it's across a little valley, we find obsidian from the Iburu complex, which would have been more than 100 kilometers away on foot. So long-distance transport of this material. What are they doing with the material? They're making small and medium-sized points that were hafted. They're making bladelets and little bladelet cores and small end scrapers. And Levallois technology is the dominant technology. It's also more than 500,000 years old at both the Alorgesailie and Wonderwerk Cave in South Africa. How do you maintain a large social network? You use pigment. And we have this and other pieces of stone that were used to grind ochre. This is the chart I published in 2000 with Sally McBurdy, and all the red lines show where we've gone since 2000 in pushing back some of these distinctions of behavioral innovations that we noted in that paper. And just to give you a line, here's the Homo sapiens first appearance datum, and you can see the lines go well back before anything we're going to call Homo sapiens. And just to show you, we also have discontinuous preservation, which could be showing us that we have these things in perhaps small packages that appear and disappear. Perhaps the people go somewhere else for a while and then they come back. But we have periods of time when we don't have a lot of evidence and periods when we do. But you can see that the record is not enormous for all of Africa. So in conclusion, I have just a few points which I'll read very quickly. Most technological innovations are older in Africa, and they imply at least 4 to 5 out-of-Africa events, not just 2. And Africa had probably consistently fairly large populations compared to Eurasia, because it's a wonderful place, particularly East Africa, to be a terrestrial herbivore. And as a friend in this audience once pointed out to me, it's wonderful to have 2 rainy seasons a year, because then if one of them fails, you have the second one to hope for. There's the Neanderthal Denisovan split from our ancestral lineage may be one such event. The onset of accelerated behavioral evolution, innovation, considerably predates the Homo sapiens in Africa and the Neanderthal split. So Neanderthals eventually share pigment use, Levallois technology, and probably cognitive capacities for language. But later Africans with projectile weapons and large-scale networks would have held a major competitive advantage over Neanderthals, and eventually, as we know, wiped them out. The discontinuous nature of the African record implies small or fluctuating scale groups with potential for extinction, especially in subtropical ends of the continent. And it also argues that ancestral morphs and artifact styles could well have survived and did survive. The second early Homo sapiens in Ethiopia is associated with the Acheulean, with hand X's. And so we also have a new archaic human that was described this past year from Nigeria that's dated to 11,000 years ago. So again, there are mysteries in Africa, always something new from Africa. And I'd just like to thank CARTA and the organizers of this conference, many of my closer collaborators on several of these projects, and the funding organizations. Thank you.
So my talk here is to give you a little insight into how, at least I think, culture influenced the evolution of human genes in the recent and perhaps not quite so recent past. So what kinds of evolutionary relationships between genes and culture might we imagine? There are several modern proposals on the table that reflect the old dichotomy between nature and nurtures that propose, for example, that culture is really largely under the control of genes and natural selection on genes. The most prominent spokesman for this point of view, I think, is Edward Wilson, who has this, who proposed with Charles Lumsden clear back in 1981 that culture was under kind of a genetic leash, that natural selection acting on genes would give us psychology that then shaped culture in ways that were dictated ultimately by the survival value for our genes. So it all boiled down to a selection on genes. The opposite kind of hypothesis is that cultural evolution at some point took over from genes in human evolutionary history, and genes no longer have anything interesting going on in terms of evolution. And this is a common idea, and it's perhaps tacit throughout most of the social sciences, that social scientists don't have to worry about genes and genetic evolution because it's all the interesting stuff is going on in culture. And The position that I defend, and I'm going to talk about here, is that the relationship is much more intimate between genes and culture, and in particular that culture often leads the gene-culture coevolution process. So cultural evolution creates novel environments, and then those novel environments exert selection pressures on genes, so that genes play, culture has played a very active role in the formation of human nature, if you want to think of it that way. So the old nature-nurture dichotomy really has to be broken down into three kinds of elements that are important in human behavior. First, genes, second, culture, and third, individual learning. So we get the direct impress of the environment on ourselves, and then we get the evolution of these two inheritance systems. Part of this depends upon the idea that culture is a form of inheritance. So it's a lot like genes in some important respects. You get your culture from other people, from your parents and from other people that influence you. Your peers, a large number of other people may influence you, but it has in common with genes that something is transmitted, that behavioral variants are acquired by imitation and teaching and spread among people, much like genes spread. But of course, there are these dramatic differences. And one of the most important differences is that culture is a form of inheritance of acquired variation. A meeting like this is all about the inheritance of acquired variation. Those of us who speak think we've learned something in our scientific work, and we're trying to teach it to you to convince you that we've learned something interesting. So we're an example right here, a laboratory example of how that system works. We can also use the much larger community of people than just our parents to acquire our culture from. So if dad is a poor hunter or mom can't weave a basket to save her soul, then we can learn from aunts and uncles and other people. And of course, we dramatically amplify that kind of thing in the modern world when formal teaching comes along, and we learn a large number of skills in formal settings by professional teachers. But nevertheless, the pattern that so struck Darwin of descent with modification is something that obtains in the cultural system as well as in the genetic system. And so culture links learning and inheritance through this inheritance of acquired variation. And the important part of that is that it means that cultures can evolve much more rapidly than gene pools. So it adds to natural selection. Natural selection may still act on cultural variation, but we add to it the ability to use learning and to use selective borrowing from other people to cause evolution to happen in a much more rapid pace. So Darwin himself was already a pretty sophisticated cultural evolutionist, and you can extract passages from The Descent of Man, like the one I have here, in which he remarks that, or it's really an opinion, that in highly civilized nations, natural selection is no longer so important, and the continuing evolution depends more upon the teaching of children and good education, he says, and high standards of excellence, depending upon the best and the brightest, to acquiring culture from the best and the brightest, and these are then embodied in laws and customs that are taught directly to children. So this, he just barely uses the word culture in the modern sense in one place, but it's full of ideas like tradition and customs that are related to the modern concept of culture. Unfortunately, The Descent of Man was an unduly neglected book, particularly after about 1900, and Darwin's influence was lost, and for 3/4 of a century or so. So how can we imagine that culture could have a major impact on human evolution, that we could be substantially made by cultural evolution, even to the point of our genes adapting to cultures that we invent? So in the first place, culture has been important for at least 250,000 years, probably more like 2.5 million years. So Allison's talk just before mine gave you a sketch of what we know about that. And if cultural evolution is faster than genetic evolution, then culture is going to often play the leading role in the evolution of this gene-culture coevolution system, not a lagging role. So we have this long period of time in which culture and genes interacted and in which the cultural system could have played a leading role. And so cultural processes create novel environments, and then such environments lead to selection on the gene. So just to reiterate the point of the previous speaker, we have this succession of fairly sophisticated and increasingly sophisticated stone tools over that long 2.5 million year longer period of human evolution. Now, so what direct evidence do we have that changed cultural environments have imposed themselves substantially on genes? Quite recently, we have made a major transformation in our environments, particularly in the elaboration of agricultural subsistence systems, which have replaced meat largely in our diet by plant materials, and we've done things like invent dairying and generated an entirely new nutritional system for most human populations that no longer depend upon hunted and gathered resources. And this seems to have led to, when we survey the genome looking for events of strong selection in the gene, something we can do, looking for selective sweeps as the jargon goes. We can estimate the age of these sweeps by the amount of material on either side of the allele under selection that have been dragged to near fixation by the selective sweep of the target gene because of linkage. So we could take it, and then this linkage breaks down gradually over time. So it provides a kind of a molecular clock to date these selective sweeps. And at least if the preliminary data that's in the Hawks and Harpening et al. Paper in 2007 is indicative that there was a huge wave of change associated with agriculture. So that's a recent, very dramatic change. There are some quite convincing cases were actually known before modern molecular genetics. So in human populations with dairying, most adults secrete lactase throughout their life and can digest the sugars in milk throughout their life. In other mammals and most human populations, this gene is shut down about the time of weaning because it's no longer useful. Only humans with dairying get much milk to drink after they're weaned. There are many hemoglobin polymorphisms that turn out to be adaptations to exposure to malarial parasites, and these polymorphisms all seem to be quite recent because human populations seem not to have been dense enough to sustain malaria as a specialist parasite on humans until the advent of agriculture. So this is another case of the imposition of an agricultural environment, in this case making humans dense enough to support diseases. Many other epidemic diseases have a similar history that came to afflict humans after the development of agriculture in more dense populations. So there's presumably going to turn out to be a large raft of these. Some of these things are much more speculative. So there's some idea that genes that affect behavior might have been selected by the advent of societies that are hierarchical. So there's a proposal that certain serotonin transporter genes that make people more tolerant of inequality have arisen in the last few thousand years as inegalitarian state-level societies have replaced the egalitarian societies of our hunting and gathering days. And then the human dispersal out of Africa was presumably led by cultural innovation. So when people penetrated from tropical Africa, subtropical Africa, into the mammoth steppe, into Europe and these other cold, even periglacial environments, we built fires and made shelters and learned to make highly tailored clothing. But in addition, a considerable number of genetic adaptations occurred as culture dragged us into these novel environments. Skin pigmentation is a well worked out one in which exposure to low UV environments, and particularly in northwestern Europe. Northwestern Europe is the cloudiest environment in the world, and so it's and light-colored skin allows people to synthesize more vitamin D. Adaptations to high altitudes. There's an interesting adaptation in Tibet in which the way in which the humans respond to low oxygen by producing fewer red blood cells than they otherwise than the rest of us. Turns out that confers health advantages on people living in high altitudes, and it's present in Tibet, but not in the Andes. And Tibet was settled 30,000 years ago or so, and the Andes not until a few thousand years ago. So the Andean people have not yet acquired that adaptation, if they ever would. A conjecture here then is that culture may have played a big role in all of the attributes of humans. And one particularly interesting one is human social behavior. So humans are docile, they live in these large groups. Allison already mentioned that. And so might humans have acquired these proclivities through some form of group selection. It occurred to people going clear back to Darwin that people look like they've been selected as tribes, that we have loyalty to tribes, we recognize a boundary of tribes, we act altruistically towards other tribal members, we behave honestly with other tribal members, not necessarily so honestly with regard to others. And Darwin thought, and again in The Descent of Man has this little passage that suggests that humans indeed are group selected. So this is just a quote. You've had time to glance at it from The Descent of Man. So one of my students a couple of years ago, Adrian Bell, got the idea that maybe we could look at the variation between human groups, the genetic variation and the cultural variation, and put it on the same scale. So the problem with selection among groups from a genetic point of view is that it's a little bit of migration tends to homogenize the genetics of two groups, and human groups are really not very different genetically, which means it's hard to imagine that the group selection process that operated on humans operated directly on genes. So there in the left of that diagram are the FSTs. This is a proportion of variance that's between groups relative to the total genetic variance in those bars on the narrow bars on the left, and you can see they're quite small. And the gray bars are the cultural variation based upon the World Values Survey, and you can see that it runs about an order of magnitude or maybe a little bit more higher than the genetic variation. So there's lots more cultural variation to work with. The genetic data is from Cavalli Sforza and colleagues' famous compendium of genetic variation in human populations. So the picture then that we have is that humans could have evolved by gene culture coevolution, and humans are essentially then became a domesticated animal, that we were domesticated by this cultural group selection process. So we still have these ancient social instincts. We tend to dominate other people. People are often selfish and they're often nepotistic. But on the other hand, we also have tribal scale institutions and attitudes that allow us to have cooperation in much larger groups. So if there was first cultural group selection and then primitive norms and institutions and the evolution of emotions like sympathy and patriotism and more docile behavior, and the evolution of symbolic group boundaries that certainly are very important in human groups, then we could exert social selection on people who deviate from the rules of society. This is something we certainly do today. If people don't obey the rules, they go to prison, and people in prisons don't have the same reproductive success as people out of prison. So there's social selection that way can be very important. So this is a picture of a chimpanzee brain and a chimpanzee testicles. The brains are on the right and the chimpanzee testicles are on the left. And chimpanzee males are really rough customers, and part of the reason is that they're, people think of human males, particularly juvenile males, as testosterone poisoned, but we got nothing on chimpanzees. There's, I can't, if any of you here are pathologists or know a pathologist or deal with human anatomy, I'd love to have a picture that, of human testicles and brains that correspond to the chimpanzee ones there. I haven't yet been able to persuade anybody that I know to do this. So the best I could come up with is this picture here. My picture here with, there's a human testicle, it's about the size of a walnut, I guess, and the human brain about 3 times the size of a chimpanzee brain. And so you can imagine what that picture would look like if I could put my hands on one. Okay, that's the end of my remarks. Thank you very much for your attention.
As we've heard from a number of talks today, we know that humans originated somewhere in sub-Saharan Africa, and from there they expanded to occupy essentially every corner of the Earth landmass. And in doing so, they have encountered a tremendous diversity of habitats and environments that differ in terms of climate, which is a major focus of our research, but also in terms of nutrient availability, resource availability, degree of solar radiation, and so forth. And these environments are illustrated here in this map of the ecoregions of the continents. And so these different aspects of human environments have exerted strong selective pressures on human metabolic processes and physiological processes, and certainly adaptations have arisen in response to these selective pressures. So environmental change is truly a defining feature of human evolution, where in part is due to movement of populations, therefore experiencing new environments, but there is also variation of habitats and environments over time, illustrated here in this diagram that has population size on the vertical axis and time on the horizontal axis. And as you can see, there are a number of changes marked by different transitions during human evolution. Among these transitions, we recognize 2 as perhaps the most important ones, meaning the ones that have probably been associated with most selective pressures. The first one is the out of Africa expansion, which occurred sometime earlier than 40,000 years ago. During this transition, humans were exposed to colder climates, lower degrees of UV radiation, different nutrients, and also lower pathogen loads. And then much more recently, sometime more recently than 9,000 years ago in the Neolithic Revolution, humans shifted away from a subsistence based on foraging, which characterized much of human existence up to that point, and the adoption of different subsistence strategies based on horticulture, pastoralism, and intensive agriculture. These changes, in turn, induced a number of other changes, for example, at the level of the diet, which became much more rich in carbohydrates, and milk became a major staple of adult diet. But also there were massive increases in population densities, which led to an increase in pathogen loads, an increase in transmission rates of infectious diseases. So really, as I said, environmental change has been a major defining feature of human evolution. And humans have encountered this tremendous diversity of environments, responding with cultural, behavioral, and genetic adaptations that led ultimately to the wonderful diversity of phenotypes and cultures that we see in human populations today. Some of the phenotypes that vary across human populations are actually diseases. In fact, many common diseases have significant interethnic differences, and these include diseases of the immune response like asthma, multiple sclerosis, but also metabolic diseases like type 2 diabetes or different types of cancers like prostate cancer. There are some phenotypes that, in addition to varying greatly across human populations, they also have striking geographic patterns. This is the case, for example, for body size and proportions, which are known based on classical work to be correlated with climate variables. As shown here on the vertical axis, we have the body mass index, and on the horizontal axis, the mean annual temperature in worldwide human populations. So that populations living at opposite ends of the climatic range, like these Maasai in eastern sub-Saharan Africa and these Inuit from eastern Siberia, have rather different body size and proportions, which suggests that humans, just like most other mammals, conform to general ecological rules that suggest that body size and proportions are adaptations to different types of climates. Another phenotype that has striking differences across ethnic groups and also striking geographic patterns is pigmentation. We know that skin reflectance, which is a measure of pigmentation, is strongly correlated in worldwide populations with latitude or distance from the equator, which, as we've heard before, suggests that variation in pigmentation is adaptive with regard to different degrees of shortwave radiation. So these are two cases in which phenotypic variation appears to be a function of a particular environmental variable. So we thought that based on this information that one could look for genetic variants that have similar geographic patterns, with the idea that these genetic variants might be adaptive with regard to different aspects of the environment. And we do that by using this approach that we refer to as environmental correlations, where essentially we look for correlations between allele frequencies and specific aspects of the environment. So we first start by classifying populations based on the environment they live in. And then we search for genetic variants whose frequency is correlated with the environmental variable of interest in the populations that we studied. And then if we find a correlation shown in this diagram here between the environmental variable and allele frequency, we infer that this is an adaptive variant with regard to environmental variation. However, if the allele frequency is not predicted by the environmental variable, we assume that this variant does not confer a selective advantage in different environments as defined by the particular environmental factors that we've considered. So we use this approach on a particular data set, which includes more than 640,000 autosomal markers, which were genotyped in more than 1,300 individuals from 61 indigenous populations worldwide, the distribution of which is shown here on this map. With regard to the environment, we use a set of environmental variables that can be broadly classified into three categories: climate variables, which are all continuous, and then ecoregion and subsistence variables, which are categorical and analyzed as dichotomous variable. The climate variables were chosen among those that are most likely to reflect the impact of cold stress and heat stress on human physiology, as well as different degrees of UV radiation. Ecoregion is, as I said, a categorical variable that mainly contains information about climate, but as I said, it uses information in a dichotomous manner. And then the subsistence variables can be further subdivided into mode of subsistence and main dietary component, where the subsistence mode includes foraging, horticulture, pastoralism, and advanced agriculture, and the main dietary components are cereals, roots and tubers, fat, milk, and meat. So we've applied this approach to this genome-wide data set, and I'm just going to show you a couple of examples. So in these two diagrams, which reflect two strong signals of correlation between a SNP and, in this case, a foraging subsistence, while in this case, it's a correlation at this SNP between allele frequency and relative humidity. What we find is that on the horizontal axis, we have the individual populations, but grouped based on the major geographic area. And then on the vertical axis, we have allele frequencies. Now, if we focus on the left portion of this slide, I color-coded the populations, or at least the categories of populations. In red, we have the foragers. In light blue, the non-foragers. And then this horizontal line indicates the mean allele frequency for populations within a certain category and within a certain geographic area. And so what you can see is that the signal that we find is that not all foragers have the same allele frequencies, and all non-foragers have a different and identical allele frequency. Rather, what we find is that there is a consistent shift in allele frequencies between foragers and non-foragers that is observed in multiple geographic regions, as if selection acted on subdivided populations in parallel to increase the frequency of a particular variant that is advantageous with regard to that particular aspect of the environment. And this SNP is in a gene that codes for interleukin-22, which is a potent mediator of the innate immune response. And so it may make sense with regard to the changes in pathogens associated with the foraging to non-foraging subsistence. On the right, we have a correlation between allele frequencies and a climate variable, which is continuous. And again, the pattern that we see is that there are clines of allele frequencies that repeat themselves in multiple geographic locations. And in this case, this is a non-synonymous substitution in the keratin 77 gene, which is expressed in the ducts of eccrine sweat glands. And therefore, it makes sense that this SNP might reflect adaptations to heat stress in influencing the way the body can cool down its temperature by sweating. Another example, in this case with a dietary component, was observed at this PLRP2 gene. This gene codes for an enzyme that metabolizes galactolipids, which are a main component of plants. And interestingly, what we found is that there is a polymorphic stop codon. So in other words, this is a shorter protein that occurs at higher frequency in populations that specialize in cereal compared to populations that do not, as shown here, where the red indicates the frequency of the variant in populations that specialize on cereals versus the blue populations. So again, these consistent shifts in allele frequencies that are observed in multiple independent locations. And the interesting thing is that we predict, based on the biochemical evidence, that this stop codon, even though it results in a truncated protein, this protein is more active, and therefore it makes a lot of sense that it's found in populations that have a plant-based diet, which is rich in cereal. Another type of analysis that one can do to learn more about the biology of these signals, but also to sort of increase our confidence that we are finding something real, is to look at whether these signals are enriched in genes that have specific biological functions. And so we looked at dietary specializations in roots and tubers, which is one of the classes where we have some of the strongest signals. And what we find is that the two biological pathways that are most enriched in these signals of environmental correlations are starch and sucrose metabolism and folate biosynthesis. And that's actually what you would expect for roots and tubers, which are rich in starch and poor in folates. And so it's very comforting to see this particular result. With regard to adaptations to the polar ecoregion, we find several metabolic pathways that are involved in energy metabolism, and therefore in the production of heat and the maintenance of body temperature in the face of a cold climate. Another type of approach that we can use to learn about the biology of these environmental adaptations is by comparing our results with those from genome-wide association studies. So as I just explained, our approach connects particular polymorphisms with specific aspects of the environments that are considered proxies for the selective pressures that underlie the signals that we observe. Genome-wide association studies, in contrast, connect polymorphism with specific phenotypes. And so now if we look at the strongest signal from our analysis, as well as the strongest signal from genome-wide association studies, and we ask which polymorphisms have strong environmental correlations as well as strong association with phenotypes, we can start to make a connection along this side of this triangle and ask questions such as which phenotypes were acted on by natural selection and which specific aspects of the environment shaped these phenotypes, which selective pressures shaped these phenotypes. When we do this exercise, we find that there are many SNPs that have strong signals with climate and are associated with pigmentation and tanning phenotypes in genome-wide association studies. This is exactly what we would have expected based on what I told you in the beginning, that the pigmentation phenotypes have this clinal distribution with a gradual change as a distance, as a function of distance from the equator, as a function of solar radiation. The other result that we have that perhaps is less expected but very interesting is that many of the overlap between the two analyses identify the phenotypes of the immune response, and in particular autoimmune diseases like multiple sclerosis, systemic lupus erythematosus, celiac disease, Crohn's disease, psoriasis, and so forth. This is also not entirely unexpected. Because we know that there is a great deal of diversity in the way, there is a very striking pattern of the geographic distribution of pathogens, which is very established in the literature, that pathogen diversity decreases as a function of distance from the equator. And it was shown a few years ago that climate, and in particular humidity, are very important factors in shaping this gradient, this latitudinal gradient of diversity. So it makes sense that we find these particular observations which suggest that pathogens are probably the true selective pressure acting on these polymorphisms, and that climate is essentially acting on pathogens, and which in turn act on polymorphism. So we are looking basically at the indirect effect of climate on selected alleles. Now, by no means these pathogens represent the only selective pressure shaping this kind of signals, but it certainly, as based on this analysis, it's certainly an important component of it. So to conclude, I showed you that there are very strong genome-wide signals of adaptations to climate, to ecoregion, to dietary components and subsistence in worldwide population samples. Many of these signals are found in genes whose function can be easily connected with the biology of the adaptations and the specific environmental factor that was used to identify the signals. The signals are due to relatively subtle, and I didn't talk much about this, but relatively subtle but consistent shifts in allele frequencies that occur in multiple geographic regions across populations that experience different environments. And we found that adaptation to different climates make an important contribution to pigmentation phenotypes as we expected, as well as to diseases of the immune response. I'd like to put a plug for this dbcline. This is a database that we have generated in my lab. You can find it at this URL, and you can search the database for signals of environmental correlations with 21 environmental variables, and we have links to other selection browsers. And then finally, I want to thank the people in my lab and my collaborators who contributed to this particular research.